Hello, and welcome back. So, what I want to do now is talk about one of my favorite groups of secondary metabolites known as the terpenes, which I'm showing you here with my cursor. I love these compounds. They smell like Christmas and they are so fun to study, which is why my lab basically focuses on terpene research. Okay, so what I want to do here is just show you some common terpenes that you may or may not already know about. So one here is myrcene, which is a very common terpene that you can smell when you crush hops in your hands. And that's the compound that gives hops that kind of musky, earthy smell. Another one is linalool that I think we're all familiar with from lavender. It gives it that kind of floral, sweet smell that we associate with those calming, relaxing benefits that we get from smelling it. Caryophyllene is a compound that is characteristic of black pepper, gives it that woody, spicy scent. When you crush rosemary in your hands, it kind of smells a little bit like pine. And that's because there are a number of pinene molecules that actually make up the majority of that chemical profile. And actually, in citrus fruits, so like limes and lemons, and oranges, you tend to smell limonene, and that is what literally gives it that citrus lemony smell that we really enjoy. Okay, so now what I want to do is just talk a little bit about the chemistry and not too much, and then we're going to hop right into monoterpenes. Okay, but terpenoids or isoprenoids in general, I just want you to know are incredibly ubiquitous. They are present in all living organisms, and they have this unusually high diversity in plants where actually over 23,000 of these compounds have been identified as plant natural products. So mainly, what we see is that these terpenoids are these cyclic compounds containing 10, 15, 20, or correspondingly more carbon atoms in multiples of 5. And this is because they're produced by adding a different number of this 5-carbon precursor molecule that I'm showing you over here, this isopentenyl pyrophosphate or IPP. So basically what happens is these just get added sequentially during synthesis. And what you can see over here for the different types of terpenes, the empirical formula always has the carbons as a multiple of 5 because of that. And obviously, synthesis is a lot more complicated than what I just told you, but you get the gist. And that's how we get all of these different molecular formulas that I'm showing you here on the right. So I just want to mention, we're going to talk obviously about plant insect interactions here mostly, but I just want to mention that terpenoids actually do play a role in primary metabolism because these compounds can function as membrane constituents. They're incredibly hydrophobic, so they can just sit right up there in that in the biphosphate matrix. They can also serve as pigments for photosynthesis. So carotenoids like alpha and beta carotene, which are actually tetraterpenes, they have 40 carbons. They are electron transport carriers, and they're also growth substances and hormones. So you may have heard of gibberellins or auxin, and these are actually terpenoids, and they stimulate stem elongation, germination, and flowering, and other kind of those benchmarks of development. Okay, but as secondary metabolites, or specifically their role when we think about plant insect interactions, they have lots of different functions. So they can serve as feeding and oviposition deterrents. They can inhibit ATP synthases. They can interfere with molting and development. They've been known to disturb the nervous system of insects. And remember, I told you they're volatile. And so as volatile organic compounds or VOCs, they've been known to serve as both direct and indirect defenses, where they can just be directly toxic, but also serve as indirect defenses where they kind of can recruit these natural enemies of herbivores. Okay, but what I want to focus on today, and let me go back one second, is I want to focus on these monoterpenoids, these compounds that are basically have the C10H16 molecular backbone. And the reason I want to talk about monoterpenes is because we do a lot of research with conifers. And when we think of conifer chemistry, I think the first thing that comes to our mind is basically resin or oleoresin. And you can see the resin just coming out here from a cut piece of wood where the resin ducts are now exposed to the atmosphere. And one of the dominant secondary metabolites in conifer resin is this class of compounds known as monoterpenes. So again, they are typically, these cyclic 10 carbon molecules. They're produced from pyruvate and G3P through a pathway known as the MEP pathway in plastids. And they're primarily found in resin ducts, resin canals, resin blisters. And you can see here, this is actually a transverse cut of a needle from a conifer. And these circles here that I'm pointing to are all resin ducts. And these resin ducts are actually lined with plastid enriched epithelial cells. You can see them right here, actually, if you can see my cursor. And those are the cells that are responsible for synthesizing terpenoids. And then what happens is they secrete them into the extracellular lumen, so into that space, that lumen space, and they just accumulate there under pressure. And because these compounds have high vapor pressures, they actually volatilize into the atmosphere, so they can make their way even through this very hydrophobic cuticle because they are hydrophobic compounds, and they can enter the atmosphere. And that's what gives conifers these lovely scents. And so while the resin itself can serve as a physical defense against insects by just flushing organisms out and entrapping them, they also serve as a chemical defense and cause insect mortality because basically they just have a general toxicity to them, and they also have a number of antifungal properties. So monoterpenes, like I said, they're found in both plant tissues, but also in the atmosphere, and can influence multitrophic interactions in a few different ways. So they can influence species interactions first from the top down. And I think Claudio may have touched on this, but I'm just going to reiterate that here for monoterpenes, because they can impact the ability of insects to basically locate their host plants for feeding. They can affect parasitism, so helping parasitoids locate their herbivore hosts, and they can influence insect oviposition choice. So they can also, though, affect these species interactions from the bottom up, right? Because they're directly toxic. They can feed as serve as feeding deterrents, sorry. And they also just affect an organism's immunity and growth. Okay, so we know that climate change is going to influence most, if not all, of the interactions I just mentioned. And the shifts in chemistry that are going to occur in response to climate change are obviously going to play a role. And so my lab specifically studies drought and drought effects on plant insect interactions. And droughts are not only becoming more frequent, but they're also getting hotter. And these drought events are highly correlated with insect outbreaks. And so, one thing that we're asking is, well, is it because these droughts and these hotter droughts are altering monoterpene concentrations? That's just one question that my lab wants to find out. And so a good example of this type of drought event or drought-induced tree mortality event occurred in the early 2000s in the Southwest, where that region experienced one of the worst droughts on record. So this is a pretty well-known photograph taken by Craig Allen in 2002. And this is the same area taken just 2 years later in 2004. And as you can see, the entire landscape in this pinyon juniper woodland has changed dramatically due to drought and bark beetles. And so what's surprising to me, I guess, is that in light of events like this, we still don't know what tree traits, including the chemistry, that make some of these individuals more susceptible than others to initial bark beetle attacks. And the question I have basically is, can we quantify kind of these physiological thresholds of plants and trees that lead to increased susceptibility to these destructive insects? In other words, at what point along a drought continuum can trees no longer make these defense compounds and therefore suffer from attack by destructive agents like bark beetles? And so, I think basically just knowing how monoterpenes and other defense compounds, how they change in response to drought stress, has the potential to help us when we're trying to understand how beetle population dynamics are going to shift in the future. And this is because monoterpenes in particular play crucial and diverse roles in bark beetle chemical ecology, including that of the pinion engraver, or Ips confusus, which I'm showing you right here. And it's Ips confusus that actually is responsible for all of this pinyon pine tree mortality in the Southwest that I'm showing you here. And you can see here, these are some pitch tubes on a pinyon tree, and these are what the galleries look like underneath the bark that these bark beetles create. So yes, okay. So what I'm showing you here is, and what I want to emphasize is that monoterpenes play crucial and diverse roles in bark beetle chemical ecology. And they play kind of these diverse roles because high concentrations of monoterpenes can be incredibly toxic, but low concentrations of these compounds can actually be used to the beetle's benefit. And I just want to give you a few examples. And the first one is with cis-verbinol. So cis-verbinol here, which I'm highlighting, is a minor constituent of the pheromones that these pinyon engravers produce. And what that does is it's a pheromone signal that basically aids in recruiting beetle friends to come to the tree and being like, hey, I found this great resource, you should check it out. And so it basically helps in creating what we call mass attacks. And what happens though is that this cis-verbinol is actually produced through the hydroxylation of alpha-pinene, and alpha-pinene is produced from the tree itself. And that is just so crazy that these insects can actually rely on its host compound and hydroxylate it and then produce a pheromone that is going to actually help it. So when we see high concentrations, this compound alpha-pinene is toxic, but in low concentrations, the beetle actually uses it to its benefit, right? And there are other compounds, other monoterpenes that are present like beta-myrcene that are produced by the tree and actually can serve as pheromone synergists and can help the beetle. But again, only at low concentrations. At high concentrations, they can be quite toxic. So there's, again, this dose dependency and this kind of fine line between toxicity and what might be beneficial to insects. And so monoterpenes not only influence the insects themselves, but can also affect the fungal symbionts that the bark beetles carry. So bark beetles actually rely on fungal symbionts to basically supplement their nutritional needs. And so it's really important that the fungi they carry to the trees can also survive. But monoterpenes affect the ability of these fungal symbionts to grow and to reproduce. And so these are just some data from a separate paper, not one of ours, that was published last year that shows that the growth of Ophiostoma ips, which is basically this blue stain fungus that's vectored by bark beetles, that it was strongly influenced by 3 monoterpenes. So you can see alpha-pinene, limonene, and myrcene. And you see that alpha-pinene led to actually a decrease in growth of Ophiostoma ips. But the presence of limonene and myrcene actually tended to enhance it, where maybe the fungus can use those molecules as carbon substrates for growth. And so you don't have to know all this, all the details here, but what I'm trying to show you is that basically monoterpenes not only influence the insects themselves, but also impact the fungal symbionts that those insects vector and can have really important repercussions for the insect's fitness and reproductive potential. So I just want to end here just by saying that ultimately to make monoterpenes, it comes at a cost, right? And so monoterpenes are made from photosynthates. So here we see photosynthesis occurring, and from photosynthesis, we have the production of what we call non-structural carbohydrates, which are basically just sugars, soluble sugars, for most cases. And the photosynthates and these non-structural carbohydrates, they can become limiting under drought stress, right? And not only that, but trees allocate these resources, these non-structural carbohydrates, to other things like storage and growth and reproduction, as well as constitutive and induced secondary metabolites, which are shown right here. And so, these terpenes, again, are some of the most costly metabolites that plants can produce by mass. And so because they're so expensive, you can expect there to be trade-offs with these other pools and processes. And that just gets really complicated when drought is involved. And obviously that gets more complicated. And then the downstream effects on herbivores and pathogens can become even more complex. And so we have lots of cool results and research coming out of the lab with regard to this kind of line of research. And so I just would say stay tuned. And, we're going to be discussing these compounds more during class in a week or two, and you'll get to smell some of them and think about the roles they play in plant insect interactions by participating in a hands-on activity that we have planned. So that'll be really fun. And yeah, I just want to thank you for listening and tuning in. And I'm excited to talk more with you about this topic. And yeah, looking forward to seeing you soon.